Eso.
Yung Smith and Brown, yung Smith and Brown classification po. Smith and Brown classification, yung alpha hemolysis partial, Smith and Brown classification.
White pinpoint colony, sir.
Espiogenes po and ah, Espiogenes po, saka S. agalactiae.
Hindi po.
No din po.
No, sir. Wala pong boyfriend, sir. Girlfriend lang po.
Naka-mobile data din daw po.
Wait lang, pauwi pa lang po galing PhilHealth, sorry.
Yes po.
Yes
Yes.
po.
Sir, may nagre-report sa akin, by the way. Wala daw audio yung YouTube mo, but rinig yung students?
Hello, 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 hello. All right. So okay, so we have this already. I can hear your names already. Okay. So yeah. So mamaya babalikan natin yan for the video, 'di ba? For the FORD. Talagang naiinis talaga ako pag narinig ko yung for the. Yung ganyan. Naiinis ako talaga diyan. Tapos ganyan pa yung spelling, capital F and capital O. Gigil na gigil ako pag nakikita ko yan sa Facebook posts. Okay? So nasa na ako? Nawala tuloy ako. Thank you so much for informing me. Whoever that is. Si Sir Marco ba yung nagsabi? Alright.
Yes po.
Okay, so nasaan ang aking ano, nasaan ang aking butihing ano, ang aking butihing what do you call this? Ang aking butihing butihing moderator. Alright, so ganon. So unidentified organism, then question sa baba, what's the organism? Right? So they will give you like they will give you a question like this in ASCPI, and you will have several choices. Okay? And then sometimes you will have a control organism here, and then a non-control organism. So you might see something like that in the exams. Okay? So tandaan nyo siya ha. Tandaan nyo, okay? So this is good practice also to study this way, because you will understand how organisms are identified in the exams. Okay? Para hindi kayo ma-surprise. Okay? Para hindi ka ma-surprise ng GF. Okay? Alexis? Alright? Wonderful. Anak ha? Anak. Makinig ka anak ha? Anak. Nako, I have a soft spot for people who are quite different from everybody else, and because I'm also like that. All right. So I love my children. All right, my kids. All right. So let's move on to our next slide. So you guys understand how we are going to differentiate the organisms. All right. Thumbs up or raise your hand if you did not understand para explain ko for person. Wala. Okay. So we can move on na for the people. For the people. All right, we can move on. All right. So in PYRA's test, so we're going to discuss these four, by the way, before we continue. These three, sorry. Bacitracin is basically sensitivity testing. All right. So in PYRA's test, which beta hemolytic strep test is positive? All right. PYRA's test, beta hemolytic strep. So again, it's Streptococcus pyogenes. Now, we're going to talk about the substrates that are being used or that you need to have in the exam. Kaya binalik ko. Ayokong makita nyo kaagad ang sagot. So the substrate that we're going to use is, of course, pyrrolidinyl aryl amidase. Okay? And the color developer is para-dimethylamino-cinemaldehyde. Don't forget, cinemaldehyde. Sarap yung cinemaldehyde, yung cinnamon. Charot, para hindi nyo maalimutan. Okay? Para amino-dimethylamino-cinemaldehyde. And the positive color, the positive result for this one is a red change of color. Clear? Clear ba? Clear, Eliza? Is it clear to you, Eliza? All right, so we can move on to the next one. All right, we can move on to the next one and indicate the color reaction catalyzed by PYRAs. So PYRAs is basically, we're going to talk about it in biochemical sense. The color reaction will change to pyrrolidinyl. PYRAs will catalyze the reaction with levopyrrolidinyl beta naphthylamide. Then it will break it down to levopyrrolidinyl and beta naphthylamide. Okay, this is what it looks like, the actual biochemical reaction. And the beta naphthylamide, the one that you're seeing at the other side here, in the left side, sorry, in the right side, sorry. Kaliwete kasi ako kaya I have some sort of minor dyslexia. Um, will react with paradimethyl cinnamaldehyde. What is paradimethyl cinnamaldehyde? You ask. It's the substrate. All right. It's the substrate. Sorry, the color developer. Sorry, the color developer. Okay. This is PYRAs. Okay. All right. So this one is PYRAs. This one is this one is the one that we are targeting. So in clinical chemistry, you already know these things. There are certain substrates that are being there are certain end products that will be detected by color developers, and your color developer, as I mentioned before, is para dimethyl cinnamaldehyde. So please don't forget that the original or the base molecule is this one. Okay, pyrrolidonyl beta naphthylamide or levopyrrolidonyl, and it splits up to this one. Okay, this is a ketone molecule, hence the term pyrrolidone. Okay, right, and this one is naphthylamide. So it's an amidic form. It has a nitrate group. Okay, clear? Clear? Before we move on, clear ba? Clear. Sinong tatanungin natin? Clear ba, Maria Cristina K. Sabado? Okay, clear for you?
Yes po.
Clear? All right, wonderful, wonderful. Okay, now we're going to move on with camp. Who here can transcribe the meaning of the camp test? It's name of four people. Camp test. Yeah. Who here knows? Okay, McKay.
Christy Atkins Munch Peterson.
Okay, wonderful. That's actually correct. All right. Peters, Atkins, Munch, and Peterson. And what is the positive result? What do you think is the positive result? Anybody? Let's go again. Roll call. Um, Okay, Nigel, all right. Hindi na ako magtingin sa chat box, all right. You guys can raise your hand. Okay, so okay, Nigel, what's your answer?
Arrowhead po.
Okay, what?
May arrowhead.
Arrowhead what? Arrowhead colonies, arrowhead hemolysis, arrowhead pinkish
Arrowhead.
discoloration, arrowhead what?
Arrowhead.
Hemolysis. Okay, but I told you specifics kasi baka mamaya tinamaan ako ng pulog, inuuto ko lang kayo. Di ba? I told you guys specifics. I need the specifics. Okay? So thank you. Thank you, Nigel. You gave me a partially correct answer, which is okay. It's acceptable. But again, it's okay to be wrong here. Um, but make sure in your exams you learned your lesson. Okay, Nigel? All right. So be specific. Baka mamaya tinamaan ako ng pulog. Sabi ni, gaganon si Segi. Ano nga, sabi ni Sir Arrow, ang naalala ko, sabi lang ni Nigel, Arrowhead lang. Wala. Ano siya, Arrowhead colonies pa yung isasagot ko. Tilalo kang nalito, di ba? Alright, so I want you guys to be specific. So what is our test organism? What's our test organism? Or control organism? Yeah. We have a control organism. Kasi may follow-up question ako after this one. I have a follow-up question. Yeah. What's our control organism here? Can't do camp tests without this organism. I told you guys that yeah, a couple of weeks ago. I think a couple of weeks ago when we discussed hemolytic patterns. You guys forgot already? The test organism. Sige, Mark Segi.
Sir, agalactiae, Streptococcus agalactiae.
Control organism. I'm talking about control organism. I'm not talking about the organism that tests positive. What about James Paul? Thank you,
Okay.
Mark. Okay, please remember. Okay, just
For useful, sir.
what?
Stuff for useful.
All right, wonderful, wonderful. Okay, so please remember that, Mark. Okay, so that's correct. So the organism, the beta hemolytic strep that tests positive, I'm going to ask it again several times para talagang kumatok sa utak ninyo, okay? Kasi marami talagang students nakakalimot. So I'm going to ask it again. I know you already know this, but girl, I already know this. Ganun yung boses ni Valiant. Very urban ng accent, di ba? Girl, you already said this. Why are you repeating yourself? Ganon magsalita si Valiant, no? Ganon, nagagalit. Sabi niya ganun. Kasi paulit-ulit. Bakit paulit-ulit, sir? Kasi baka makalimutan ninyo. So, strep agalactiae will test positive with this one. Okay? Alright, strep agalactiae. Who's the one that tests, which one of the two will test positive with PYRAs, Streptococcus pyogenes? Which one would test positive with bacitracin resistance test? It's Streptococcus pyogenes. The remaining two, camp test and hypurate hydrolysis, will test positive with will be Streptococcus agalactiae. All right. So now, what are the reagents that we need for hypurate hydrolysis? I think I mentioned it a couple of weeks ago. All right. So please, 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 please don't forget sodium hypurate and ninhydrine solution. Now, the positive result for this one is a purple color change, right? Purple color change. Okay, and the organism that will test positive, or the beta hemolytic strep that will test positive, is, as I mentioned before, I know, sasabihin ni Valiant, girl, you already mentioned this. Why are you always repeating yourself in an urban accent? All right, in a very urban accent. Again, I'm going to repeat it. I'm going to repeat it regardless of what Valiant says. Streptococcus agalactiae, because I want you guys to keep on remembering. All right. So I know it sounds very counterintuitive that I repeat myself, but I want you guys to remember it because this is going to be part of your algorithm. If you're making algorithms, that's your way of studying. Why not? Right? Now, let's move on to the biochemical reactions that occur when you're doing hypurate hydrolysis. Sir, but kailangan namin talagang tandaan yung ano. Kailangan namin tandaan ang biochemicals. Yes. It's important you guys understand the principles because when you're doing troubleshooting and when you're understanding troubleshooting questions, you need to know what is affected, and you need to know what is going to cause interferences, especially if you are going to be asked troubleshooting questions. So magtatanong din ako noon, which of the following color developers will be mostly affected by by reagent by that interferes with ninhydrine solution? O, di ba? Kaya ganon. Nakakaloka, sir. Babalik ta rin yung question. Ganon kayo magtanong. Yes, that's how I ask questions. I want you guys to understand the principles and not just memorize random facts. It's a good thing for you guys to remember everything, but I want you guys to have an understanding or a base understanding of everything that we're speaking of here. All right, so you need to understand that the end products that are going to be measured, that are that occurs during purate hydrolysis is benzoic acid and glycine. Okay, benzoic acid and glycine. Now, let's go back to camp test. Reverse camp. I mentioned reverse camp a couple of weeks ago. All right, who here is still familiar with reverse camp? I mentioned it just a couple of weeks ago. Um, reverse camp is useful in the identification of what microorganism. We're going to stray away from our topic for a while in this particular cue card. Right? This is another gram-positive organism, but this time it's a rod and it's anaerobic, or it's a bacillus, or a bacilli organism that tests positive with camp. All right, James Paul,
Mechanobacterium hemolyticum po.
Clostridium,
Mechanobacterium hemolyticum po.
Fecalobacterium. All right, Fecalobacterium is the new term. Yeah, actually, very
Yes.
good. But what's the traditional or the board exam name pa siya? Hindi pa siya tinatanong as Fecalobacterium. But yeah, you are kind of correct. What's the previous name of Fecalobacterium? Who here knows? It's the causative agent of,
Corynebacterium.
huh?
Corynebacterium po. Hemolyticum.
Nope. Try again. It's a spore former. There's only two spore formers that we mentioned last week. Maria Cristina K. Sabador, McKay.
Clostridium perfringens po.
Okay. All right. Wonderful. Correct. So in the exams, please remember reverse camp is useful in the identification of Clostridium perfringens. We're not using the contemporary name still because I've never seen it in the exams appear in the board exams. Just a nice to know question. But thank you, John Paul, for identifying. Now, what is the modification that happens instead of using instead of using Staph aureus? What is our control organism in this case? That's actually what what is being asked in this question over here? What's our control organism or our known control? Kaya nga siya tinawag na reverse camp. Okay, Alexis.
Streptococcus agalactiae.
All right, wonderful, wonderful, wonderful. All right, Streptococcus agalactiae or agalactiae. All right, very good, very good. Okay, so what's the positive result? Reverse camp. Who knows what's the positive result? Mark, for the win, Mark. What's the positive result?
Arrowhead po.
Arrowhead
Arrowhead,
what?
hemolytic pattern.
All right, okay. All right, Nigel, did you remember? Do you remember now? All right, okay. Thank you, Nigel. All right, I mean, sorry. Thank you, Mark. All right, now sulfomethoxazole trimethoprim sensitivity test is used in the differentiation of what Landsfeld group Streptococcus. So actually going to be Strep C or Group C strep, right? Group C strep. By the way, let me just ask you again question. Let me just ask you again the question. What's the basis or the basic principles of group of the Landsfield classification? Just a recall question from our previous um from our previous discussion earlier. Was discussed 15 minutes ago specifically. So actually the first slide in this lecture. What's the basis of how do we know it's group C, or how do people or scientists came to the conclusion that this is a group C organism, this is a group A organism, group B organism? What's the basis? So you guys forgot already. The first slide is what? The first slide? What's the first slide? It's based on Alexis.
Based on hemolytic pattern.
Wrong. That is Brown classification. Next, another person. Eliza.
Landsfield classification.
Yeah, I know it's Landsfeld classification, but what's the basis? What's the basis, Eliza? Where is it based from? Mark Segi.
Sir, classification of antigenic group specific cell wall polysaccharide.
All right. Kung hindi mo sinabi yung polysaccharide, wawarlahin kita. Okay? Okay? Wawarlahin kita, ha? Nandiyan na yung clue, oh. Kayo talaga, nagagalit si Sir Marco sa inyo. Binalikan lang natin yung question ninyo kanina. Kayo talaga. Alright. Alright, anyway, let's move on. Let's talk about scarlet fever. Walang kabastusan to, ha? Alam ko yung susunod na slide. Wag kayong etchos dyan, ha? Kayong dalawa, ha, Mark? Mark. Tsaka, nasa na yung kapartner mong isa pa? Lalaki. Sino pa yung isa? Nakalimutan ko yung kapartner mong lalaki yung tumawa last week. So, pero, sino yung isa? Sino pangalan na nakalimutan ko? I forgot the guy's name, the other guys. I'm so sorry.
Yan yata si Salyan.
Sino?
Tumatawa si Salyan.
Si Arnie. Naalala ko na si Arnie. Arnie, ha. Huwag kang tatawa. Pero the reason why I always ask this question, and I always start the topic of test for scarlet fever in a somewhat derogatory way, is because I want you guys to remember. Okay, so please don't forget Schultz, Charlton, and this test. All right, so we can't say that since we're recording. All right, since we're recording, I'm not going to say it. Right, but the first test is based on susceptibility, and the other test is based on neutralization. Okay, so what is being tested by the first test? It's going to test the destruction of the bacterial cell. The inactivation of the cells is being tested by Schultz-Charlton. So I'm not going to ask you guys how it's done, because it's usually not asked in the exam. Yes, that's why I did not read it. Okay? All right? That's why I didn't read it, because The last time we discussed this, I think with the class with Mrs. Magpulak, people were laughing. People were laughing when I said it. That's why I didn't. That's why I know in YouTube on YouTube this is going to be we're going to be demonetized if I said it. All right. So next we're going to move on with tests for scarlet fever. All right. So we're going to go back. Oh, which of the two tests for scarlet fever is diagnostic? Which one is diagnostic? Okay. Is it the susceptibility one or is it the neutralization version? All right. So it's actually the awful naming one. All right. So it's the other name for this one, by the way, is the erythrogenic test, and the positive result is rashes. Okay. The other one is Schultz-Charlton, which is antitoxin. You just see blanching phenomenon, and it's actually it's actually the diagnostic test for Schultz-Charlton. And usually it's done in nursing homes. Sorry, not nursing homes. It's usually done by nurses, but it's a wonderful way for you guys to understand this. Okay, so neutralization, it happens within the body, so you don't do it in the multimedia. Okay, so nice to know. Okay, infections associated. Let's go with the infection or the pathogenesis of microbial analysis, or sorry, the pathogenesis of the microorganism Streptococcus pyogenes. What are the infections associated with um, Streptococcus pyogenes? Okay. Who here can give me one? Who here can give me one? Yeah, Eliza.
Scarlet fever.
All right, wonderful. Okay. What about Alexis? Alexis was the second one. Sorry, Marie, McKay, sorry. Okay muna. Okay. Sorry.
Yung infection po.
Strep throat. Okay. What about Alexis?
Acute glomerulonephritis.
Okay, that is under the another one of those. Okay, so I'll help you guys out. Is that the so the first two were correct. Alexis's answer was a little bit too specific. Advanced mag-isip si Alexis, which is okay. It's commendable. Don't worry. It's still correct. It's actually just sore throat or strep throat. Skin infections also, scarlet fever, and of course post-streptococcal infections. This is known actually as sequela. Okay, from one side, so it went from it started as strep throat, it became untreated strep throat. The organism went to other places around the body. So acute glomerulonephritis, if the organism produces antibodies um, that will target the kidney cells, okay, or human kidney cells. It goes to the heart also. You get you get heart diseases also with this one. So post streptococcal infection. Now, sequela. So we're talking about sequela, all right? So inunahan tayo ni Alexis. Very good anak. Very good. Very good anak. Kasi talaga, ganyan talaga mga anak ko. Pagka anak ko talaga smart na, pretty pa. Nasaan na yung isa kong anak? Nawawala yung isa kong anak. All right? Gusto ko yan. All right? Anak number one. Si anak number two, tsaka si anak number one, ha? Pangalawa ka kasi ikaw yung second child, okay? Hindi ikaw yung hindi ikaw yung mag-inherit ng throne, girl. Sorry. Ikaw lang yung ikaw lang yung maggagawa ng kabulastugan sa throne natin. Alright? Talagang Game of Thrones ang reference, tsaka House of the Dragon. Alright? Game of Thrones ang reference and House of the Dragon. So, nasa ang anak ko na isa? I want my other child to answer this one. My first child, my firstborn. Wala. My firstborn is still struggling with internet. My God. O sige, yung ampon ko na lang. Sige, si Mark Segi na lang. Yung ampon ko. My third child.
Examples of
Examples of sequela. I just mentioned it earlier. Acute glomerulonephritis and rheumatic fever. So you guys forgot already rheumatic fever, right? So sequela. Rheumatic fever occurs when antibodies directed towards streptococcus antigens are directed towards the towards glomerular epithelium or what cardiac muscle tissue. Okay, so be mindful of these things. Okay, Now, pathogenic determinants. So we're going to pathogenic determinants. Streptococcus pyogenes. We have a special. This is actually just a discussion, a specific discussion on Streptococcus. All right. So there are a lot. Sir, tatandaan namin lahat yan. Yes, that's a lot. Hemolysins, streptokinase, streptodomase, diphosphatidine dinucleotidase, erythrogenic toxin, polymerases like DNases and RNases, and the M protein. Sir, parang familiar sa akin si M protein. Yes, you are not mistaken. You're not mistaken, James Paul. I can tell that you're a little bit overwhelmed by the pathogenic determinants. But yes, these are the pathogenic determinants, James Paul. Okay? So hemolysis, you already know it breaks down cells. Streptokinase is the hyaluronidase version of Streptococcus, for Streptococcus rather. Okay, so the hyaluronidase. Okay, so it's basically the same thing, but it's released specifically by Strep. Okay, streptodomase, clumps platelets, diphosphatidine dinucleotidase is a WBC killer, and erythrogenic toxin release causes the redness in scarlet fever. Polymerase is DNase is basically with the name itself, it destroys nucleic acids. Right? So M protein was already discussed. Who here can remember what is the action of M protein? Ge, magbibigay ako ng pera. Who here remembers the action of M protein? I want it specific. Ayan na si Nigel. This is it, Nigel. Ayan na talaga pag sinabing pera, lumalabas talaga. Anak! Nagmana ka talaga sa akin, anak! Anak! Biglang with camera pa. Kanina walang camera. Anak, ba't ganun? Alright. O sige, si Nigel muna. Sorry, anak ha. Wala akong favoritism, anak. Although you will inherit the Iron Throne, anak. Don't worry. Okay, so Nigel, ano na? Tagal naman.
Inhibits po yung bacterial phagocytosis.
Very good. Oh my God. Send me your emails. By the way, James Paul, I forgot to mention the reason why you're not being sent your prize is because you haven't been sent your prize is because my secretary in the Philippines is off. Okay, she's on vacation leave, so I can't send it to you right now. Maybe expect it 2 to 3 weeks. Okay, when she comes back to work. Okay. So Nigel, also you. Okay. So please send your regards tuloy. Ano ba yan sasabihin ko? Very Game of Thrones talaga mga references, Sir Manuel. Ano ba yan? Send your email address. Send your number here. Also say Poras, gmail.com. Okay? So yan. Alright? mdeleon164@gmail.com. Alright? Now, let's go to the actions of M protein. Very good. So she said that clumps platelets. So akala ko meron ng streptodomase. Actually, if you look at the specifics, it's actually a byproduct or a breakdown product. Streptodomase is actually a breakdown product of M protein, but I included it here. It prevents WBC chemotaxis, it prevents phagocytosis, and it has a fibrinogen-like action. What's a fibrinogen-like action? What's a fibrinogen action or fibrinogen-like action? Basically, it becomes the analog, or in the absence of fibrinogen, M protein actually causes the cells or causes the platelets to clump. Okay, that's why it has a fibrinogen-like action. So clumping of platelets is an action. Fibrinogen is also an action because essentially it leads to clumping of platelets. If it doesn't have this kind of action, platelet clumping will not happen. Okay, now hemolysins produced by strep pyogenes. Okay, hemolysins that are produced by strep pyogenes. Last time somebody was able to answer. This is a question that I asked from one of Sir Marco's students. Um, Unfortunately, nobody was able to answer it, so I gave the person a chance to answer it for a prize. But since I gave it to Nigel already, I don't think we need to talk about it a little bit more. Hemolysins that are produced by Streptococcus pyogenes includes your SOS, or um, sorry, SLS and SLO. Sir, ano yung SLS tsaka SLO? Is Amanda here? Because she really likes a lot of mnemonics. Is Amanda still here? Um, Amanda, are you there? I remember Amanda, she always likes asking for, how do I memorize it, sir? Meron ka bang mnemonic? Naalala ko si Amanda last week eh. Amanda ha, you are growing on me like a fungus. Charot. Like a like a fungus. So don't forget SLO and
Okay.
SLS, Amanda, all right? Amanda, all right?
Yes po, sir.
SLO is streptolysin O. Okay? Streptolysin. S is streptolysin S, SLS. Okay? So sir, ano yung SLO? Paano ko madaling maaalala? Pag sinabi mong SLS, streptolysin S, di ba? Dagdagan mo pa ng S, SLSS, SLS, lahat ng may S. Kasi siya yung oxygen stable, Amanda. Alright? Don't forget, this is the one that is oxygen stable. That's the hemolysin that is oxygen stable. Now, ano naman yung isa? Obviously, it's oxygen labile or oxygen deterrent. Okay? Or it gets deterred by oxygen. It gets destroyed, or this hemolysin becomes oxidized. Therefore, it's rendered inactive. Right? Hemolysin O, streptolysin O, and streptolysin S. Okay? Clear, Amanda? Paano maalala kung sino yung oxygen stable? SLS lang. Dagdagan mo lang ng isa pang S sa dulo. SLS. Oh, 'di ba? Walang kakwenta-kwenta, Amanda, pero at
SS.
least maalala mo, 'di ba? Streptolysin O indicate its oxygen stability. So stability 'yon. Right? So this is the answer, all right? SLO, all right? Streptolysin O is oxygen labile. And ito nga yung pinapaalala ko kay Amanda. SLS, streptolysin S, is oxygen stable. Dagdagan mo lang ng extra S. I know walang kakwenta-kwenta, Amanda, pero at least naalala mo siya. Alright? Okay? Okay. Now let's move on to hemolysins. Hemolysins produced by strep is considered to be toxic to what cells or tissues? The labile type. Right? So this one is actually much more deadlier than this one, or much more problematic clinically. Alright? Why? Because it's toxic to heart tissue or myocardial cells and blood cells. Okay? So myocardial cells. Kaya ano ba ang target niya? Yung hemoglobin. Okay? Pero totoo, may hemoglobin din pala ang ano. No, hindi hemoglobin. Myoglobin ang target niya. Okay, so ang target niya yung mga globin chains kasi. Alright, so it binds to globin chains. That's why any cell with globin on it, not excluding globulin, is going to be destroyed by the SLO. Okay? All right? Clear, Nigel? Clear? All right. Let's move on to the next one. Inoculation technique used to identify if there is oxygen labile hemolysins in clinical isolates. Actually, this is an edited version of you guys are lucky because people from my previous class did not know this. It's actually stab and streak. Okay? SLO strains will lyse the blood in the stab area. So, anong itsura nun, sir? Gusto naming makita. Gusto niyo pakita via ano? Gusto niyo pakita ko via actual practice? Kasi nandito naman ako sa lab. Gusto niyo yon, 'di ba?
Yes
Gusto
po.
niyo? Or ayaw niyo? Bahala kayo kung ayaw niyo. Sige, ano? Gusto niyo, guys?
Gusto po.
Ay, totoo. Eliza. Nasaan yung anak ko? Yung anak ko wala. Ginagawa natin lagi sa bahay, no? Yung dalawa kong anak, ginagawa namin lagi sa ano, yun yung family bonding namin kung paano ginagawa yung stab and streak. Yun yung, before makapagsalita si Valgat, yan yung una kong tinuro sa kanya. Charing. Charing. Alright. Wait, kukuha ko ng aga. Tingnan ko kung gagana ba. Gumagana ba ang ano? Webcam. Alisin ko muna yung webcam dito sa ano. Kasi parang nag-interfere siya sa ano. Tapos, ayaw. Ayaw maano ng webcam. Ayaw eh. Webcam. Para walang pictures, di ba? Walang pictures. Ano eh? Bakit ayaw? Wait, let me just close this one. Ay, Ako pala yung nawala. Sorry naman. Video with integrated camera. Wait. Let me. How do I do this? Pero pag may mga test tayong gagawin, papakita ko sa inyo in actuality so you guys know, you guys will understand. Video. Magulo ang buhok ko sa video. Sorry, guys. Sorry. Wait. Chill. Bakit ayaw niya? Ayaw niya mag-share. Ayaw niya mag-share. Anyway, let's just talk about it some other time. Paalala mo sa akin, Segi, ha? Please remind me later. Right? Ayaw niya mag-share. Gusto niya dun lang sa may streaming app ko. Nakakabwisit siya. Anyway, let's continue na. Am I sharing the screen with you guys now? Let's just talk about it later. Please remind me later, Mark, what about
Apo.
it. Okay? So before we go on break, when we go to Neisseria species. All right. So this is the one. All right. So there you go. Um, am I sharing my screen now?
Yes po.
All right. Okay. So next question is so I told you guys about stab and streak. I'll show it to you guys later. Let's go with the neonatal infections commonly associated with beta hemolytic strep. So neonatal infection. So certain neonatal infection are usually associated with to what or with what dapat siya. So I made this presentation when I was still a student. I was just updating it every time. So mali-mali pang grammar ko diyan, guys. Huwag kayong itsusera diyan. Okay? So bata pa ako noon. Mga kasing tanda ko pa si Minty noon nung mga panahon na 'yon. Okay? Medyo medyo hindi lang ganoon kalantong. Okay? Hindi lang katulad ni Minty kasi medyo demure ako noon. Mga itsura ko noon dati katulad pa ako ni Arnie. Yung kala mo lalaking lalaki. Ganon. Alright? So neonatal infections are commonly associated with what? Beta hemolytic strep. So we only discussed two hemolytic strep, beta hemolytic strep. What's the answer for this one? Arnie. Kala mo Arnie nakalimutan kita ha? Naku, nakalimutan na ni Arnie. Sige. Steel. Valiant. Ayan na, anak. Ano na, anak?
Streptococcus agalactiae.
Alright, tatama pa ang pronunciation. Medyo ano lang sa dulo. Nautal lang sa dulo. It's Agalactiae, anak. Okay? My gosh, natutuwa talaga ako kay anak. Ayan na, anak. Di ba? Talaga ngayon talaga ang ano ko, heir to the Iron Throne. Alright? The heir to the Iron Throne. The Becky Throne. Alright? Okay, now, group D Streptococcus is divided into two types or two groups. There's actually two groups. We have the Enterococcus species and the non-Enterococcus species. We're not going to dwell deep into this one, but I just want you guys to remember Enterococcus, and the Enterococcus group species include the following organisms: Enterococcus faecalis, Enterococcus faecium, Enterococcus durans, and Enterococcus avum or avium. Okay? And the non-enterococcus species include, or the non-enterococci, the organism that does not grow in, or that are not classified rather as non-enterococci, are Enterococcus bovis and Enterococcus equinus. All right. So for Enterococcus species, you can remember that all of this comes from the poop. Okay, from your poop. Okay, so it could be poop from birds, could be poop from humans, and could be mixed poop from everybody. But the non-Enterococcus group comes from what? Bovines, diba? Right? Cows and equines, which is horses. Okay, so see? Galing, sir. Ang dali lang pala niya maalala. O, 'di ba? Para hindi kayo makalimot. Ang dami na nating pinagdaanan ng mga mikrobyo, no? So sana maalala niyo lahat. Lagay lang ng lagay para anyway, hindi naman ako mag-aaral niyan. 'Di ba? Yun yung sinasabi ng ano, yun yung sinasabi ng isa sa mga review center ano namin. Review center lecturers namin dati. Sabi niya, lagay lang ng lagay, class. But anyway, hindi naman ako ang magre-review niyan. Ganon. All right. Now, we need to differentiate group B species. Group D. The Landsville group D. So you need two culture media need? You need two types of culture media. Okay? So you need BEA. Sorry, you need two components of it, by the way. Sorry. Components of this media. By the way, the culture media that you need to use to differentiate group D from other Landsfeld group species is bile esculin hydrolysis. Okay, bile esculin hydrolysis. Si Bea. Huwag mo kakalimutan. Amanda ha, huwag mo kakalimutan another acronym. Bea. Alright? Amanda, nandyan ka pa ba? Are you still there, girl? Are you with us? Move the glass if you can hear
Yes,
me.
yes, sir.
Alright. O
Still
gumalaw,
po.
nakakatakot. Nag-Ouija board ako dito, biglang gumalaw si Amanda. Ganun. Huwag mong kakalimutan, Amanda, si Bea ha. Hindi Bea Alonso. Bile esculin agar ha. Huwag Bea Alonso. Alright. Now, the components of this one includes 40% bile and esculin. Alright. Now, we're going to move on with the reaction that occurs in bile esculin agar. So, the reaction that occurs is that there is a formation of ferric chloride. Okay, there's a reaction of ferric chloride. Ferric chloride reacts with the esculitine or the breakdown product or esculitine, the breakdown product, and it turns this from a yellow or it turns this from a yellow test tube to a yellow culture media rather to a black culture media. When you see things like ferric chloride or ferrous ions, tandaan nyo, nagbablack lagi yan. Okay? Ganito yung itsura niya. Credits to Tom, sorry, credits to Tom Barlman. Okay, 'di ba? So talagang kailangan natin ng citations lagi. Ha? Mark, huwag mo kakalimutan ha. Nag-citation tayo. Pag hindi ako nag-citation, Mark, paalala mo sa akin. Because you are my moderator. We have to make
Yes, sir.
citations. This is from a random medical technologist that I just Googled. Okay? So nilagay ko dyan talaga from Paul Kemer. Alright? From Tom. Sorry, from Tom Barman. Okay? So, huwag niyo siyang kakalimutan. Barlowen. Sorry, baka mamaya masingil tayo ni YouTube. 'Di ba? Ganun. Okay? So, let's move on. What tests may be used to differentiate two species of, Enterococcus species from non-Enterococcus species? We can use growth on 6.5% NaCl and PYRAs. Okay? Or PYR test. Pyrrolidonyl, Keme. Okay? You just use PYRAs, the PYRAs. And I know what's the what's the substrate that we use for PYRAs, for PYR test, and what's the color indicator? Sige nga. Baka nakalimutan niyo na naman. Ano? PYR muna tayo. PYR kasi isa sa mga test dyan. Kasi there's two tests. Sige, who can remember what's the reagent? Ano, ibabalik na natin ulit sa PYR. Guys, sana kakasabi ko lang kanina. Ayan na si Alexis. Anak, number 2.
Dimethyl amino ay dimethyl sin
Bilang lola.
na
Anak.
dimethyl aminocinnamaldehyde.
Okay. And what's the color? That's the what? Is that the substrate or the
A
color
substrate.
developer?
Substrate.
Mali.
Ay, sorry,
Anak.
sir. Color developer.
Color developer siya. Alright. So, what's the substrate? Sige, Mark Segi. Bumalik si Mark Segi sa previous lecture natin. Binalik niya yung YouTube. Perks of being the moderator. Sige.
Pyrrolidonyl arylamid
Arylamidase.
arylamidase.
Alright. So, please don't forget those two things. What's the positive result? Anak, the heir to the Iron Throne. What's the positive result of PYR? Anak, hindi ka talaga nakikinig, anak, ha? Disappointed ako sa iyo ngayon, anak.
Pink formation.
Ha?
Pink or red formation. Di ko sure.
Anong bubble formation, anak?
Pink
Anak,
or red.
Catalase yun, anak. Ikaw talaga, anak, ha? Catalase yun. Somebody raised their hand before Nigel. Eliza ata yun. Eliza, what's the positive
Red.
result? Red color, the development of a red color indicator. Alright, very good. Anak ha, wag katalis ha. Anak ha. Anak, sasampalin kita. Hindi kita pinalaki ng ganyan, anak. 'Di ba ang ABC mo nga, ABC ng mga bakterya, 'di ba? Aerobacter, Bacillus, 'di ba? Yun nga ang ano mo, 'di ba? Yun ang ABC mo, Aerobacter, Bacillus, Clostridium, 'di ba? Si Alexis lang nga ang hindi na biyayaan kasi tinamad na ang mother. Sir, pink or red, sinagot ko. Parang may narinig ako formation eh. Akala ko bubble formation. Ikaw talaga, anak number one, ha? Tatanggalin na kita as heir to the Iron Throne. The Iron Becky Throne. Alright, so okay. So very good. Now, which group tests positive for both? It's of course this Enterococcus species. The growth on 6.5% NaCl. Sir, meron din growth on 6.5 NaCl, sir? Sino ba yung organism na is capable of growing in high salt concentration? Who's the other organism that we talked about? Again, recall questions. These are just recall questions, so please be mindful of these ones. Sige. Last week lang natin diniscuss. Magagalit sa inyo si Sir Marco. Last week, staff grows on 7.5% NaCl, guys, 'di ba? Kayo talaga, ha? You guys forgot na, 'di ba? Ito naman 6.5. Ito naman, kayo talaga, ha? Anak talaga, ano na talaga kita. Disappointed ako sa performance mo ngayon, anak. Anak, ha? Wag ganon. Mark, hindi mo ata nababantayan to, hindi mo ata to nababantayan this past couple of weeks. Hindi nag-aaral ngayon ata to anak ko. All right, next. Let's go with the alpha hemolytic streptococcus. I mentioned it before, but tatanong na naman ni Valiant, girl, you already mentioned it before. But in a very urban accent. Urban accent, di ba? Girl, you already mentioned this before. Alpha hemolytic streptococcus. Brown classification. Answers. Anybody? Alexis.
Streptococcus viridans, Streptococcus pneumoniae.
All right. McKay, what's the answer to the other one? We have 5 tests to differentiate this group, and which group tests positive? Which is the 5 what are the 5 tests? Very important. I saw it in somebody's algorithm. I don't know if that person remembered it.
Sir, tatlo lang naalala ko.
Sige lang, go. Push.
Yung optosin sensitivity, new fluid key lung test, and yung bile solution po.
Okay. Very good. Very good. Very good. Very good. Kalimutan mo ang animal inoculation. Traditional test kasi siya. Animal inoculation, optogen sensitivity, bile solubility, inulin fermentation, and Neufeld Kilang test. Okay? Neufeld Kilang test. All of this will test positive for Streptococcus pneumoniae. Alright? Streptococcus pneumoniae. Alright? Clear? Clear? Who are what age group is affected by Streptococcus pneumoniae? I discussed it a couple of slides ago and last week also when we were discussing I think the differences between Haemophilus, right? What else? Haemophilus, yeah. So, what age group? Nakalimutan na nila. Nakalimutan na talaga nila. Sir Marco, nakalimutan na nila. Patay. Parang ginaganahan akong pahirapan tong mga estudyante na to. Ginaganahan akong pahirapan. Pag-ising-ising lang si Mark Segi kasi ang dami niyang blast. Pag-ising-ising lang siya. Alright. So what's the age group? Neonates or newborns. Okay? Ay, naunahan na kita. Sorry anak number 2. Oo. Gusto ko yan. Yung mga ano, yung one paragraph or two paragraph yung ano, yung case ng patient. Then identify the isolate. Di ba? Tapos may picture pa, Sir Marco, para panlito. All right, now let's go with Amidace. Let's go with Amidace. Sorry, where were we? Nasaan ako? Nawala yung slide ko. Ayan, let's go. Test used to determine the presence of amidase. It's actually bile solubility. Please don't forget this. There's a difference. Bile esculin and bile solubility. A lot of our students from the previous batch always made mistakes when it comes to bile solubility and bile esculin. These are two different tests. Okay, so bile solubility is different. You use sodium desoxycholate. Okay, you use sodium desoxycholate, which is the bile salt that you use. All right, and we use muramic acid. Muramic acid reacts to dissolve colonies. Okay, so after addition of sodium desoxycholate, the muramic acid as an end product and alanine will actually dissolve the colonies. Okay, so this is what happens. All right. Okay, so bile will be dissolved by sodium desoxycholate will be dissolved by your by muramic acid, and then it will cause the dissolution of the colonies. Okay, all right. So other name for taxo P. Oh wait, let me just pause here for a couple of seconds because I have. All right, let me just pause for a couple of seconds. I want you guys to answer. Send your answers in the chat box. Can you guys give me 15 minutes? 5 minutes. We have an emergency sample. Right? I want you guys to give me the answer for um for example, my answer for first bullet like this. Answer. Okay? Answer 2. Answer. Okay? So I want you to follow that format, okay? I'm giving you guys 5 minutes. I just need to run a sample, okay? And I'll show it to you guys later, okay? I'll give you guys the correct answer. I want you to type it in the chat box, okay? In the chat box. All right. Hello, everybody. Okay, so what happened? What is our answers, guys? Alexis Valbona did not follow the format. Maria K. Sabado did not follow the format. Nigel kind of followed the format, so okay. Let's go with Nigel's answer. So Nigel said optochin. Let's see if she's actually correct. Optochin. All right. And then the other name is what? Sorry, the chemical name is ethyl hydroxycuprine hydrochloride. Wow. Ethyl hydroxy. It's supposed to be ethylene. Okay. So optochin sensitivity test, ethyl hydroxy. It's ethylene hydroxy. Ethylene hydroxy. Very good. Eliza was actually the only one who answered it correctly. Everybody used ethyl hydroxy. Ethylene. Ethyl. So it's actually Eliza. Eliza was actually. Congratulations to Eliza. Actually, wala namang prize yan. Wala lang. Nag-break lang ako eh. May run kasi akong sample. Okay? Wala namang prize yan. Pero give her a hand, guys. Come on, guys. Come on. Can you guys clap for her? Yeah, yung clap na, yung katulad nung clap ni Valiant. Di ba yun? Di ba yun? Ayan. Over naman to si James Paul. Over naman to si James Paul. Nakakaloka ka, James Paul. Natuwa naman ako kay James Paul. Ganun pala yung clap ni James Paul. Alright, now, let's move to the most sensitive test used for the detection of this is the sorry, the most sensitive test used for this is the most sensitive test used for serological detection of strep pneumoniae. Right? This is the most sensitive test, the most serological, the most sensitive serological test dapat. And again, I was a child when I read this. You guys just edit the grammar, okay? To detect Streptococcus pneumoniae. This is sinabi na kanina ni McKay. Pangalan siya ng dalawang tao. No, sige, McKay.
Neophid kilang po. Neophid
Alright,
kilang test.
very good. Nufeld kilang. Or what's the other name for Nufeld kilang?
Scapular swelling po.
Capsular. Kulang ang
Ay,
specifics.
capsular.
Remember, I need specifics. Remember, bakit? Ano bang pinagkaiba kasi ni S. pneumoniae with other Streptococcus species? Kasi siya lang ang may capsule. Right? And, yeah. So, what are the reagents that we need to use? We need anti-capsular antibodies and we need methylene blue. Okay? Anti-capsular antibodies and methylene blue. And of course, the reason why I didn't write the positive test is, of course, you already know it. Positive test. Sorry, the positive result for the Neufeld Kilang test is, of course, capsular swelling. So why would I bother writing the positive test? You guys have so many things to remember already, right? Okay. Ayoko na dagdagan nyo na ako ng dagdagan, sir. All right. So what is the other term used for nutritionally variant streptococci? Nutritionally variant streptococci. Anybody knows? Anybody knows? I think this was a question in my board exam. Which of the following organisms is, which of the following genera? Yeah, in my board exam, this was a question. Which of the following genera is a nutritionally variant streptococcus? All right, so Ira Joy. Sorry, I thought she raised her hand. Sorry. Who knows the answer? Unmute yourself and show yourself on camera. James Paul.
Abiotrophia po.
All
Tsaka
right, very good. It's actually,
granulocycaltella.
yeah, very good. I love it. It's actually a Abiotrophia species. Actually, the pronunciation is incorrect, but okay, but that's okay. At least you have an idea. It's a Abiotrophia. All right. So under Abiotrophia, previously these are the organisms that were known as now known as Abiotrophia. Before it was Streptococcus adjacent and Streptococcus defectivus. Okay, and both of these organisms require vitamin B6 for growth. Okay, pyridoxal. All right, vitamin B6 for growth. Now it's known as Abiotrophia adjacens, Abiotrophia defectivus. How do they know this? They use what you call, sorry, nucleic acid sequencing, and it's the R16 RNA 16 or RNA 16S analysis. Okay, we'll talk about that later when we go with nomenclature. This is how microorganisms are identified nowadays. Okay, or taxonomically classified. So you guys remember when you were studying biology, I think in your second year in high school, or I'm not sure kung but when you were in high school, you have what you call taxonomy, right? Taxonomy. Remember what's taxonomy, right? Taxonomy, right? Anybody knows what a taxonomist does? It's a branch of biology that is responsible for classifying organisms. Now, the current trend, or rather, not the trend, the contemporary way of identifying or classifying organisms nowadays is through RNA 16S, okay, which is a universal identifier for microorganisms, specifically bacteria. Right? We'll discuss that later when we go to, I think, a discussion in Mycobacterium, because there's a new edit in the correct nomenclature of the proper organisms there. So we'll discuss it further. All right. So just chillax and listen to my lectures, and we'll go to that part of microbiology in a couple of days or weeks, or in a couple of weeks. Okay. Now, the growth requirement is vitamin B6 for abiotrophia. All right. Now, dental caries. Ayan na naman tayo. Binigyan ko na, actually, dinagdag ko yata to ngayon linggo lang. Nung nabasa ko to. Kasi I remember nga si Amanda. I remember Amanda. She likes to remember, um, she likes she likes, or she reviews using mnemonics. Alright? So, of course, baka magtampo yung ibang students na nagre-review through mnemonics. Pinagbigyan ko naman si Amanda. So, ayan. Streptococcus mutans, mitis, sanguis. Huwag mo kalimutan. Laruin mo lang yung SMS. O, di ba? MMS naman ngayon. O, M&M's. Di ba? M&M's. Huwag mo kalimutan. Streptococcus mutans, Streptococcus mitis, and Streptococcus sanguis. Ito yung nagkukos, by the way, pala guys, ng bad breath. Kapag maraming mga sira-sira sa ngipin, yan yung maa-isolate mo ng mga organisms doon. Okay? Huwag yung kakalimutan, ha? Alright? Now, Streptococcus species associated with brain and liver abscesses, right? Sanguinosus, intermedius, and miliary. Hindi ko, kinaklasify din ba siya, sir, with that? Huwag kayo mag-alala. Alright? Huwag kayo mag-alala. Usually, ang questions is, which of the following organisms or strep species or organisms under the genera Streptococcus is not associated with liver and brain abscesses? Ganun mga tanungan dyan. Aalisin nyo lang select which one does not belong. Pero ako kasi, ang ibibigay ko sa inyo, huwag kayong charotera dyan. Ang ibibigay ko sa inyo is cases. Okay? Yung isang one whole sheet of paper ang question. Tapos apat lang na questions ang follow-up. So pag hindi mo talaga na-identify, sorry ka na lang. Pero ang hindi sorry is si Mark Segi. Kasi moderator. Ganun talaga pag tinanong ko sino gusto mag-volunteer as ganon. Sagot agad, di ba, Mark? Ganon. Kahit hindi mo alam kung saan ka kung ipagkakanuno ka ba ni Sir Marco, no? Iaalay na pala yung kaluluwa mo sa demonyo. Hindi mo alam. Di ba? Oh, yan. All right. Now, let's move on with another condition associated with Streptococcus sanguinosus is, of course, aside from brain and liver abscesses, is your subacute bacterial endocarditis. Okay, subacute bacterial endocarditis. It's been isolated in many cases, especially for heart surgery patients. Okay, so that's the end of our discussion about Streptococcus species. We're now going to go with Neisseria species and other gram-negative cocci. All right? Are you ready? Are you ready, guys? Or do you guys need a break? What, Mark?
Sir, pa-break po muna. Mga
Okay, very good.
15 minutes.
Alright. Ba't parang galit ka?
Hindi po.
Charing lang. Kala ko ganun na. Sir, break muna. Ganon. Alright. So, how many minutes would I give you guys? 15 minutes?
Yes po.
Alright.
10
Sige.
or 15 minutes po.
15 minutes na lang para ano. Pero guys, pwede ba if I would ask, like 15 minutes din mag-extend lang ako mamaya? Kasi di ba na-late ako ng
Okay po.
pasok? Okay lang yan. Okay lang ba yun kay Sir Marco? Baka may klase pa si Sir Marco eh. Wala naman. Hindi nagsalita si Sir Marco, so I guess that's okay. Anyway, I'll see you guys in 15 minutes. Wait, start ko yung timer para fair. Okay? Baka mamaya sabihin nyo unfair ako, bigla na lang ikaw dumating, Sir Manuel. Anong kaguluhan yan? Okay? So, start ko yung timer. May timer ako dito. Share ko sa screen ko
Okay po.
para alam ninyo talaga. Okay? Baka mamaya para may idea din kayo. Papasok-pasok lang kayo. Okay? Share screen. I know, share window. Share window. Nasaan yan? Wala. Entire screen na lang para makita nyo din yung mga mukha nyo. Okay. Ayan, nakita nyo?
Yan pa lang, sir.
15 minutes ha. 15 minutes. Walang daya-daya to ha. Pag wala kayo. Hala na wala. Pag wala kayo, bahala kayo sa buhay nyo dyan. Okay?
Yes, sir.
Alright. Alright, here you go. I'm gonna stop the stream.